Hi, I am Dr. Arsalan and today we are going to discuss the structure of nephron. Nephron is a microscopic structure. It is the basic unit of structure and function of kidneys. Each human kidney is made up of 1 to 1.5 million nephrons. If we elaborate the structure of nephron, it comprises of basic two components, the renal corpuscle and renal tubules. The renal corpuscle, this segment constitutes renal corpuscle and the rest of the segment is renal tubules. In case of renal corpuscle, there is tuft of capillaries called glomerulus. Glomerulus is constituted by the efferent arteriole. Basically, as we have discussed in our previous lecture, that the hilum of the kidney, in case of kidneys, this is the hilum where the renal artery innervates. This renal artery branches and rebranches and forms efferent arterioles. These efferent arterioles, these form a tuft of capillaries called the glomerulus. The glomerulus is enclosed in a cup-shaped structure. This cup-shaped structure is called Bowman's capsule. As it is evident in this diagram, that this Bowman's capsule is made up of two layers. So this is a double-layered cup-shaped structure. This is also a microscopic structure. It encloses the glomerulus in it. The cells of the Bowman's capsule which are surrounding this glomerulus, these cells are called podocytes. These have keen role in blood filtration, which we will discuss in our next lecture. So this was the renal corpuscle, the segment comprising glomerulus, Bowman's capsule and podocytes. This is called renal corpuscle. The second structure is renal tubules. Apart from this renal corpuscle, all these structures are contained in the renal tubule segment. Renal tubules have three components. The first is proximal convoluted tubule. This is the proximal convoluted tubule, the first segment, which is emerging from the Bowman's capsule leading to the loop of Henle. Second is the loop of Henle. This is second segment, loop of Henle. This is U-shaped structure, starting from this end and ending at this end. This point is called descending loop of Henle, while this region is called ascending loop of Henle. And the last portion is distal convoluted tubule. Third part, it is called distal convoluted tubule, which again rises towards the Bowman's capsule and opens into the common collecting duct. This collecting duct collects urine from different nephrons. So different nephrons opens in this segment. The glomerulus leaves the Bowman's capsule by efferent arteriole. This efferent arteriole, this coils around the tubular part of the nephrons, especially around the loop of Henle. This coiling is called peritubular capillaries. Peritubular capillaries are the microscopic blood vessels which emerges from the glomerulus in the form of efferent arterioles coiling around the tubular part of the nephron. This is called peritubular capillaries. And if this coiling of peritubular capillaries occur around the loop of Henle of this nephron, this is called vasrecta. So these are two basic nominations of the blood vessels which are emerging out of the glomerulus of the nephron, that when the glomerulus is left by the efferent arteriole and it is coiled around the tubular part of the nephron, it's called peritubular capillaries. And if this coiling occurs around the loop of Henle, this is called vasrecta. This loop of Henle is of very much importance in case of reabsorption of water, which we will discuss in the function of nephrons. So in case of cortical nephrons, the loop of Henle is short. The peritubular capillaries are curved or coiled around the proximal convoluted tubule and distal convoluted tubule. While in case of juxta medullary nephrons, loop of Henle is long as it is shown here. So in the juxta medullary nephrons, the peritubular capillaries wrap around the loop of Henle, forming vasrecta. The prime function of vasrecta is reabsorption of water from the loop of Henle into the blood vessels, so that the body water level osmoregulation is maintained in effective way. So the channel of blood vessels across the kidneys and nephrons is divided into 6 steps. First of all, the renal artery enters the kidney through the hilum, as we had discussed in our previous lecture, that the renal arteries enter the kidneys through hilum, this notch. This renal artery then branches and rebranches, forming efferent arterioles. The efferent arterioles further splits and divided in a coiled form lying in the Bowman's capsule called glomerulus. This is the tuft of capillaries, and the glomerulus leaves this Bowman capsule via efferent arteriole. The efferent arteriole emerging from the glomerulus when coils and supercoils around the tubular part of the nephron form peritubular capillaries, and if it is coiled around the loop of Henle U-shaped part of the nephron, this is called vasrecta. And then vasrecta, these combine and drain into the renal vein. This is the renal vein. So vasrecta and peritubular capillaries, these combine and drain into the renal vein, and renal vein emerges out of the kidneys through this hilum. So this constitutes the vascular system of kidneys and nephrons.